So after such many kind of the microscope, now is very interesting, and my favorite microscopes is the transmission electron microscopy. It is really very interesting kind of the microscope, and you can see the images in the 3D and 2D images are also. The transmission electron microscopy is also called TEM microscopy, a technique that uses a beam of the electrons transmitted through the specimen and the ultra-thin section of the specimen and image on the imaging device. So in this kind of the microscope, a beam of the electrons will be used, which will be transmitted from the specimens, and the specimen should be really ultra thin. The specimen should not be a little bit thick or the other kind of the special kind of the specimen. The specimen, there is special procedure to make the specimen for such kind of the microscope. The principle lying behind is the focused beam of the electrons from an electron gun passes through the ultra thin section of the specimen. As we have already studied, that specimen for this kind of the microscope should be ultra thin space section, and the beam of the electrons, which will be the focus and will be passes through by an electron gun, and beam gets focused on a small area of the specimen and by an electromagnetic condenser lens. Instead of being placed on a glass slide, the specimen is usually placed on a copper mesh grid. This is also one difference with the other kind of the microscope, that the specimen will not be placed on the glass slide. It's better the specimen is placed on a copper mesh grid. The illustration of the transmission electron microscopy, the thermocouple gauge, cold cathode gauge will be there, and the specimen port on the right side, column roughing valves, desiccator inlet, diffusion pump and rotatory pump will be on the left side. Similarly, the air inlet, camera valve, they are self-explanatory image of the transmission electron microscopy. You can also call it as TEM microscopy, TEM transmission electron microscopy. As we have already studied the image formation, electron passes through the specimen and then through an electromagnetic objective lens, which will magnify the image. The electrons are focused by an electromagnetic project lens onto a fluorescence screen or a photographic plate. The final image, called the transmission electron micrograph, appears as many lights and dark areas. We will see the image on the later slides, depending on the number of the electrons absorbed by the different areas of the specimen. So the specimen, the electrons will be absorbed, will be passed through the specimen, and the specimen is dependent on how many electrons are passing through that specimen or through that area of the specimen. That's why we have already said that the specimen should be ultra thin specimen. As the thinner specimen, they will be able to absorb more and more electrons, and it will be give us a more better picture under the microscope. The transmission electron microscope can resolve the objects as close together as 10 picometer. The objects are generally magnified 10,000 to 1 lakh about. The contrast, the dye used for such kind of the transmission electron microscopy, the contrast can be greatly enhanced by using a dye that absorbs the electrons and produces a darker image in the stained region. There are some different kinds of dyes which are available and which can be used. The lead, osmium, tungsten and uranium stains. These metals can be fixed onto the specimen with the positive staining, are used to increase the electron opacity of the surrounding fields, which will be in the form of the negative staining for the TEM. Limitations of this kind of slides are the only a thin section of the specimen, because we need the ultra thin specimens for such kind of person. There will be also the struggle and the labor will be used to make it the ultra thin specimen, and only the ultra thin specimen of the some special object can be studied very effectively under this kind of the microscope, which should be about 100 nanometer, not more than 100 nanometer. The specimen must be fixed. They need to be fixed. They should be dehydrated and viewed under a high vacuum to prevent the electron scattering, because we are using the electron gun to pass the electrons through the specimen. These treatments will not only kill the specimen but also cause the shrinkage and distortion. So it is very carefully such kind of the microscopy should be done, because we are already giving it the very ultra thin microscope, ultra thin specimen need to be used, and then it should be the fixed and it should be dehydrated and it should be viewed under a high vacuum to prevent the electron scattering. So therefore there is need to be very careful under such kind of the circumstances, so that the specimen should get alive and we will be able to see some better kind of the object.